Hello, I am Tahaa Saeed and welcome to our course Biochemistry Techniques. This is our first lecture और इसमें हम बात करेंगे कि हम ये subject that is called Biochemistry Technique हम इसको क्यों पढ़ रहे हैं. So basically the reason for studying this subject is कि nowadays we are studying different research articles and we are studying different subjects but we don't know the basic concept of the techniques which are being used in that article. फॉर एग्जाम्पल अगर हम एक आर्टिकल पढ़ते हैं जिसमें हमें ये बताया जाता है कि हमने एक जीन के ऊपर पीसीआर किया, उस जीन को, उस डीएनए के पार्ट को, उस फ्रैगमेंट को हमने पीसीआर के थ्रू एम्पलीफाई किया और बाद में उसको हमने क्लोन किया इन डिफरेंट वेक्टर और फिर उस वेक्टर को देन we have expressed the protein and then we have purified that protein using different techniques such as chromatography, such as maybe dialysis, such as different types of chromatographies, and then we have tested or we have checked that or detected that protein using western blot. अब ये सारी जो बातें मैंने की हैं, that are constantly being using in that these words are constantly being used in different articles, but students are nowadays unable to understand कि असल में ये बात क्या की गई है यहाँ पे. So अब आपको अगर वेस्टर्न ब्लॉट के बारे में बताया जाए तो आप जस्ट जाके गूगल करें कि वेस्टर्न ब्लॉट क्या होता है. इट विल गिव यू a note of what a western blot is and then it will give you a diagram of how we can perform the western blot, but it is not enough to understand the technique. So इस कोर्स में हम क्या करने जा रहे हैं, हम ये करने जा रहे हैं कि हम वही उन सब टेक्निक्स को जो कि नॉर्मली आर्टिकल्स में यूज़ हो रही हैं, नॉर्मली डिफरेंट प्रोजेक्ट्स में यूज़ हो रही हैं, जो कि बायोकेमिस्ट्री, केमिस्ट्री, बायोलॉजी, कुछ जेनेटिक्स की टेक्निक्स जो कि आजकल रेगुलरली यूज़ हो रही हैं इन डिफरेंट प्रोजेक्ट्स इन डिफरेंट डोमेन्स, हम उनको उनकी बेसिक्स को स्टडी करने जा रहे हैं. So I start this lecture with this introduction of the our course. तो जैसा कि मैंने आपको बताया कि it is we'll be studying different techniques to the very basics and why we are using such techniques in our labs and in different and why different researchers are using those techniques in their different projects and different research articles. Okay, so the first thing is the introduction to biochemistry. So we know that biochemistry is a subject that is interdisciplinary. So we have biology and we have chemistry, and we have joined them together to form this subject, which is called the biochemistry. And we have many other subjects of which are interdisciplinary, such as biophysics, uh, biochemistry. Nowadays, computational biology is a very, very good and very, very advanced subject that has been taught in many universities. And I myself have studied computational biology once in my life in my MPhil. So, biochemistry is a subject that deals with both the principles and the techniques of biology and chemistry. So, what we're going to do is we will be going to study the techniques that are used in biochemistry. So, some of those techniques are mentioned in the subsequent slides of this lecture that we'll discuss shortly in today's introductory lecture. Okay, so let's move forward. अच्छा, तो कुछ टेक्निक्स हैं जो कि यहाँ पे बताए गए हैं, इनको I have to try to explain a little bit to you. So the first technique is homogenization. So homogenization is used as the word is telling us. Homo means one and genization means to change it. Let's say to change or to generate something or to change its let's say form or its let's say state. So homogenization is कि हम अगर let's say कि आपके पास एक solution है that has some solid stuff that has been put it into a let's say liquid stuff. So this is the solid and this is filled with some liquid. तो आप homogenize जब आप homogenizer use करके इसको homogenize करेंगे तो after some time it will change its form into the liquid state. So all the solid matter that has been settled down into the test tube will let's say submerged or mixed with the liquid in such a way that you will get one single state of matter. अब ये टेक्निक हम कहाँ पे यूज करते हैं? We'll have a separate lecture, a separate part of lecture on these techniques. So we'll take some examples. However, I will take one example to explain why homogenization and where homogenization is used. So, for example, if you are let's say dissolving a cellular pellet in the media, you are dissolving a bacterial pellet in the media, in the let's say lysis buffer to lyse, to break the cell wall of the bacteria. You use this technique that is called as homogenization. So you have to mix the pellet, the bacterial pellet, into the lysis buffer. So you have to use very high frequencies to mix that specific mixture. So you use this technique that is called homogenization. So the next technique is the centrifuge, and let me just clear my screen. Okay. So the next technique is the centrifugation. We'll also have a separate lecture on this centrifugation technique. It is basically spinning. Why do we use spinning? So, so we use spinning because, for example, if we are dealing with, let me just turn on my pointer again. So, for example, this is a very high spinning of a specific thing, of a specific substance. So why do we use this centrifugation? It basically uses centripetal force and centrifugal force to separate an object from another object. For example, if we have some solid particles submerged or very much dissolved in a liquid that are very difficult to separate, what we do is we will try to, for example, this is an Eppendorf which has some solid particles submerged in a liquid. So what we will do is we will try to centrifuge it. And after centrifugation, you will see that all the solid particles will settle down and all the liquid will come above into the Eppendorf. So आपके जो solid particles हैं वो सारे नीचे बैठ जाएंगे और liquid सारा ऊपर आ जाएगा, and you can easily pour the liquid out and you'll get the solid particles settle down into the bottom of the Eppendorf. तो आप इजीली बाय यूजिंग सेंट्रीफ्यूज आप इजीली किसी भी सॉलिड और लिक्विड को सेपरेट कर सकते हैं. यू कैन इवन सेपरेट टू लिक्विड्स एज वेल. सो इट इज बेसिकली अ सेपरेशन ऑन द बेस ऑफ डेंसिटी. तो इफ अगर आपका एक लिक्विड दैट इज ऑफ हाई डेंसिटी and mixed with the liquid that is of lower density, you can just centrifuge them and the liquid with the higher density will settle down. जो liquid जिसकी density ज़्यादा होगी वो नीचे बैठ जाएगा और जिसकी density कम होगी that will come upwards into the Eppendorf. You can just pour it out. You can use different techniques such as micropipetting to let's say aspirate the upper layer, and you can separate the both layers into different Eppendorf. So centrifugation is a separation technique that is very commonly used in different let's say fields of biology and biochemistry. So moving on to the next technique, I'm gonna clear my screen again. So the next technique is chromatography. So chromatography is very vastly used in different fields of science, in chemistry, in botany, in zoology, in biochemistry, and many more. So इसकी बहुत सारा types होती हैं और हम इसकी काफ़ी सारे types को बारी इन इस course में study करेंगे. So paper chromatography is used to separate chlorophyll, that will study further in the slides, and TLC, that is thin layer chromatography, that uses sometimes different slides which have silica covering on the top, that is used for separate different liquids of different sometimes different pigments. और column chromatography में हम एक column use करते हैं that is used to separate different things. The next is gel filtration. In gel filtration, it is a type of column chromatography as well, but somehow different. Gel filtration में क्या करते हैं कि we use a gel that has size different size particles, uh, different sized particles that are attached inside the gel. अब होता क्या है कि हमारे जो substance या हमारा जो analyte that is used to be analyzed, वो जब उसके ऊपर pour किया जाता है तो different they are separated on the basis of different sizes. So अगर जिसका size बड़ा है that will take let's say कि depends on our gel. So जिसका साइज बड़ा है वो पहले आ जाएगा, जिसका साइज छोटा है वो बाद में आ जाएगा, या जिसका साइज छोटा है वो पहले आ जाएगा, जिसका साइज बड़ा है वो बाद में आ जाएगा. दैट डिपेंड्स ऑन हाउ वी डिजाइन our gel filtration column. Okay, the next one is the ion exchange chromatography. So in this column, the ion exchange column, the name very much tells us about that there has to be something related with the ions. So we have two ions, a positive ion and a negative ion. So if we make a column that is, let's say, पॉजिटिवली चार्ज, और हमारे पास जो हमारा एनालाइट है दैट इज फॉर एग्जांपल positive and negatively charged, positive negative positive negative, and we have to separate the substances on the base of their charges. So हम अगर इस सॉल्यूशन को इस कॉलम में से पास आउट करेंगे तो इसमें सिर्फ वो चार्ज अटैच होगा दैट इज ऑपोजिट टू द चार्ज ऑफ द कॉलम इटसेल्फ। तो जो नेगेटिव चार्जेस हैं वो, लेट मी जस्ट चेंज माय पॉइंट कलर। So जो नेगेटिव चार्जेस हैं दैट विल बी अटैच टू द कॉलम, द नेगेटिव चार्जेस, और जो पॉजिटिव चार्जेस हैं दैट विल गो out of the column. So what we did here is the positively charges or the positively charged molecule will be separated in a different tube, and the negatively charged molecules will be separated in a different tube that will be eluted from the column. We will tell that in next lectures. So these are the techniques. This is how ion exchange chromatography is used to separate the molecules which are differently charged and placed in a and are mixed together in a tube. Okay. So our next technique is the affinity chromatography. So even the name tells there has to be something reacting in this type of chromatography. Affinity means to attach something, the attachment of something, the ability of something to attach to another something. So in affinity chromatography, what we do is we design a column in such a way that it has biologically speaking, it has a receptor of something that is present in our एनालाइट। मतलब कि हमारा जो सब्सटेंस है जिसको हमने सेपरेट करना चाहते हैं, फॉर एग्जांपल कि ये हमारी ट्यूब है एंड इसमें देयर आर, लेट्स सपोज अ प्रोटीन ए, इट हैज, लेट्स सपोज अ प्रोटीन ए, अ प्रोटीन बी, प्रोटीन सी, and we have to separate the protein A. We have to separate the protein A. So what we do is we design our column in such a way that it has the antibodies for the protein A. I'll just write it as PA minus. So हमारे कॉलम के पास antibodies हैं for protein A. When we pour this solution through the column, तो protein B and protein C ये दोनों तो इसमें से पास होते हैं, and protein A will attach to the column, will attach to the column, will attach to the column, and protein A will be separated from protein B and protein C. We can design another column. हम एक और column design कर सकते हैं और हम उसमें एंटी PB प्रोटीन जो कि एंटीबॉडी फॉर प्रोटीन बी हैं उनको लगा के हम प्रोटीन बी को प्रोटीन सी से सेपरेट कर सकते हैं। एंड एट द लास्ट ऑफ दिस एक्सपेरिमेंट वी विल हैव 3 different tubes that contain PA, PB and PC separately, and we have separated these 3 proteins. We have completely separated these 3 proteins in 3 different test tubes.